Hello students, today we are going to study the DNA. DNA is a deoxyribonucleic acid. DNA is made up of monomeric units called as nucleotides. Thus, the polymer is known as polynucleotide. A fifth carbon sugar, a nitrogen containing base attached to the sugar, and the phosphate group constitute nucleotides. In early 1950, Rosalind Franklin and Maurice Wilkins used the X-ray crystallography to discover the DNA structure. Using the Franklin data and their studies, James Watson and Francis Crick established the double helical structure of DNA in 1953. According to Watson and Crick, the basic building block of DNA is the nucleotide, which consisting of three parts: a sugar molecule called as deoxyribose, a phosphate group, and nitrogenous base. What is DNA made up of? According to Watson and Crick, the basic building block of DNA is a nucleotide which consisting of three parts: sugar molecule called as deoxyribose, a phosphate group, and nitrogenous base. Deoxyribose. What is deoxyribose? It is a 5-carbon sugar molecule that forms the backbone of DNA strands. The deoxyribose sugar molecule are connected to a phosphodiester bond, creating a sugar phosphate backbone. Second, phosphate group. It is attached to fifth carbon of the deoxyribose sugar. The phosphate group is responsible for linking adjacent nucleotides together through phosphodiester bonds. The phosphate group gives a DNA a negative charge. And third, nitrogenous bases. In a cell, there are 4 types of nitrogenous bases: adenine, thymine, cytosine, and guanine. These bases project from the sugar phosphate backbone into the interior helix and involve in the complementary base pairing. Then this DNA, which is having a particular characteristics, it is double-stranded helix having antiparallel strands, having hydrogen bonding between DNA strands and complementary base pairing. Double-stranded helix, the two strands of DNA double helix coil around each other due to ATGC base pairing. Antiparallel strands means what? The two strands of helix run in opposite direction, meaning that fifth end of the one strand is paired up with the third end of matching strand. This is referred to as antiparallel orientation, and it is important for the copying of DNA. Third characteristics of the structure of the DNA, it is having hydrogen bonding between DNA strands. The base of the both strands are joined via hydrogen bond, forming AT and GC base pairing. This hydrogen bonding two bases in the base pair in the same plane perpendicular to the helix axis. The base pairs are rotated by 36 degrees with respect to the each adjacent pair in the such way that helical turns has 10 base pairs. How many base pairs? It is having 10 base pairs. Double helix of DNA is 2.2 nanometer in a diameter, that is a 20 angstrom. In the nature, DNA molecules are mostly found as right-handed helix. This helical structure results in two external helical grooves, a deep wide one and shallow narrow one. The former groove is major one while latter is minor one. And this is having a length of 34 angstrom, width of 20 angstrom. 34 angstrom is the length and 20 angstrom it is width. And 4 characteristics of this DNA, it is having complementary base pairing. For instance, if you know that the sequencing of one strand is 5-AATTGGCC3-, the complementary strands must have sequence of 3-, that is opposite. A opposite of A, it is a T, it is a base pair with a T. So T, T, A, A, C, C, G, G, 5-. This allows the each base to match up with its partner. Next point is about what is the function of DNA. DNA is necessary because it contains the instructions for an organism to grow and reproduce. Here are essential functions of DNA. Store and transmit the genetic information, that is inheritance. The primary function of DNA is store and transmit the genetic information from the parent cell to daughter cell. DNA contains the instructions that determine an organism characteristics such as its physical traits, behavior, metabolic processes. These genetic instructions are passed from parents to offspring during reproduction, ensuring species continuity and trait inheritance. It helps to synthesize proteins. One of the most crucial functions of DNA is its role in protein synthesis. Proteins are essential molecules that perform the various functions in the cells and organism. DNA serves as template for the synthesizing proteins through a two-step procedure, transcription and translation. Next lecture we will study DNA complementary strands and its transcription translation process, which involves the formation of RNA. That is the main structure of RNA and its function of RNA. Thank you.